Namaste students. Kusha Vidya. I, JP and Dr. Vitamin, and I'm Assistant Professor Jyoti VIDYAPEETH Women's University, Jaipur. In today's session, we are going to discuss about the phenotype variability, that is the penetrance and the expressivity. Sometimes we have seen that identical genes will produce different expressions patterns. Why this happens actually? What are the genetics consisting of examination of the patterns and the expressivity of the genotype and their phenotypes. So here the dominance relationship between the alleles for a given trait can be the impact of the phenotypes, okay? Or the ratio or by the interaction between the different genes can be the impact of the phenotype. So, when we say such traits that are the results from the interaction among the multiple genes and their environment, and is called the complex traits. So, given a specific traits, how can we tell whether it is a complex? Okay? So, one of the way or to recognize the complexity of the inconsistence of the inheritance or a pattern in successive generations. Okay. So here, when we say the concept of penetrance, okay, we are studying the relationship between the genotype and the phenotype of it. Okay, that means it is important to examine the statistical occurrence of the phenotype in a group of which is known as the genotype. In another words, if you understand this, that means in a given group, for example, in a given group of known genotypes for one trait, how many identical genotypes shows the related phenotypes? Okay, you might be surprised to learn that of some traits the phenotypes might not occur as often as the genotype. For example, sees everyone in a population. We carries the same allele combination for the certain traits, yet only 85% of a population actually shows the phenotypes of such expectations, the allele in a combination. So the proportion of the genotype that actually shows the expected phenotypes is called the penetrance. And thus the preceding examples, the penetrance is 85%. The value is calculated by looking at a population whose phenotypes we know. So here the large population studies are necessary for measuring the penetrance, and studies the penetrance will help, okay, to predict how the traits will be evidented, okay. So here you can see a specific examples of a complete. So here we have to see a specific example. If we are seeing in a specific example in a penetrance, an incomplete penetrance in a human bone disease, right? Or osteogenesis imperfecta, where the majority of the people with the disease have dominant mutation in one or the two genes. That means the type 1 collagen and okay, that is called 1A1 and called 1A2. That means the gene in collagen is a tissue that strengthens the bone and the muscles and multiply the bone body tissue. So here the people with osteogenesis will imperfecta will have the weak bone of bluish color and in the white, and there are and a variety of afflictions that cause the weakness in the joint of the table. That means, however, these diseases does not affect everyone who has call 1A1 and call 1A2 mutation in the same way. In fact, some people can carry the mutation but have no symptoms. Okay. So incomplete penetrance examples where even the monogenic diseases have been predictable and express the pattern in a population is actually expressed. Okay. So here the expression or the expressibility where the individual with the same genotype can also show different degree of same phenotypes or expressibility on the degree of which the trait expressed is different among the variability or not static variability among the population of a genotype. For example, the features of the Marfan syndromes vary widely. Some people have only mild symptoms such as the tall and thin with long, slender finger-like structures. So here the another examples of the expressibility at the growth is the occurrence of the extra toes or a polydactyl in cats. The presence of extra toes or the cat paw is the phenotypes of the emergence forming that of the cat or the interbreed in a generation. Okay. So we have to see that the penetrance in genetics is the proportion of an individual carrying a particular variant or an allele or a gene. That means that express or associated the traits in the medical genetics. So in penetrance, the disease is carried by the mutation is the proportions of the individuals with the mutations and have who exhibit the clinical symptoms and the individual with such as the mutation. So, for example, we can see if a mutations of the gene responsible for that autosomal dominant disorders has 95% penetrance. That means the 95% of those with the mutation will develop the disease while the 5% will not. So here the condition of the most commonly inherited or the autosomal dominant manner is said to show the complete penetrance or if you can say right, the clinical symptoms of it may be present. So here the disease is caused by the mutation or a condition which shows that the complete penetrance of the neurofibromatosis or even every person with the mutation in the gene will see the symbol or a condition. That means the penetrance is 100%. For example, the common examples we can show here or see that there are several reasons for it. That means high penetrance alleles and the high heritability symptoms are easier to demonstrate, and because if the alleles are present, the phenotypes is generally expressed. That means the allele which is highly penetrant of the more likely to be noticed in the conditions. Okay. So when the penetrance can be difficult to determine the reliability or the genetic disease caused by it, or the heritability or the onset of a system of the age related, that means age related cumulative frequency. That means the penetrance is often expressed as the frequency of a disease or a different age. For example, the multiple endocrine neoplasm type 2. So here the environmental modification or the gene modification or the epigenetic regulations take place. The assessment or here the risk for the polygenetic characters which affects the expression. Okay. So when we see the expressibility, penetrance versus the expressivity. So the expressibility is the genotype not expressed uniformly in a phenotype. All eggs are blue, but the exact shade of the blue varies with the individual with the same genotype. That means the variable expressibility is frequently attributed to the environmental factors or the variations at the gene design that elsewhere in the genome. If the expressibility or the penetrance both were right, or when maybe the continuous gradient of the phenotype between the white and the blue egg. Okay, so that means the lack of penetrance can be considered as the extreme and expressibility, in which the range expressed includes a non-expressed. Okay. Thank you. This session is part 1, 2.0 Jyoti Vidyapeeth University, Jaipur. If you have any queries, we'll discuss in the problem solving session. Namaste.